Neurological Disorders, Lesson 3.2, How do our neurons communicate with each other? The goal of this lesson is to introduce the process of synaptic transmission, which is how one neuron communicates with another neuron. At the end of this lesson, students should be able to describe how neurons change the electrical signal of the action potential into a chemical signal at the synapse, order the steps of synaptic transmission, and predict the effects of modifying the steps of synaptic transmission. We'll achieve these goals with a model of the pain pathway and a card sort of the steps of synaptic transmission. To prepare for this lesson, you'll need to review the key scientific concepts presented in it. They include the types, locations, and relationships between the different neuronal signals, including that dendrites generate electrical input signals, axons generate electrical signals, and the presynaptic terminal generates chemical signals. Synaptic transmission, which is the process of converting electrical signals of the action potential into a chemical signal at the synapse, and this concept requires an understanding of voltage-gated calcium channels, calcium-sensitive proteins, synaptic vesicles, neurotransmitters, and receptors and ion channels. You can review the scientific content in the background reading provided to you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in the lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of lesson structure, including instructions on how to manage the discussion and activity, and the student workbook provides additional explanations for your students. To prepare for this lesson, you'll also need to be sure to print the lesson worksheet and the synaptic transmission cards for your students. You may also want to have clay available for your students to build models. Students can also draw these models, so clay is optional. The key points of the lessons due now are to review neuronal structures, specifically that neurons are separated by a space called the synapse. Also, the do now serves as a formative assessment on how your students think the electrical signal of the action potential crosses the synapse. We'll get to these key points by modeling the pain pathway and brainstorming mechanisms to allow the electrical signal to cross the synapse. In the do now, which is part 1 of the lesson worksheet, students work with a partner to model the neurons involved in Sleeping Beauty being able to feel the pain of pricking her finger. Once the students model the pathway, they then brainstorm ways of changing the electrical signal of the axon into a chemical signal at the synapse. This is usually quite challenging for students, and that's fine because we'll address exactly that question today. The key points of the discussion are to review the types, locations, and relationships between different neuronal signals, as well as to introduce synaptic transmission and the characters involved, including voltage-gated calcium channels, calcium-sensitive proteins, synaptic vesicles, neurotransmitters, and receptors and ion channels. We'll get to these key points with a Socratic discussion. The discussion starts with a review of the do now. You'll use slide 3 to facilitate a discussion about your students' pain pathway models. Since the pain pathway is not the focus of today's lesson, it's used to provide a connection from yesterday's lesson to today and to get students thinking about the communication between neurons. You want to keep this part of the discussion brief and focus on the fact that there is more than one neuron involved and there is a space between these neurons. You'll then use slide 4 to discuss student mechanisms to convert the electrical signal along the axon, the action potential, into a chemical signal at the synapse. After discussing students' ideas, you'll use slides 5 and 6 to introduce synaptic transmission and the idea that there are chemicals called neurotransmitters in the presynaptic terminal that are released after an action potential has been fired, and that these neurotransmitters cross the synapse and bind to receptors on the postsynaptic neuron. From there, you'll tell the students that we'll discuss the process of synaptic transmission as if it were a play, with characters performing actions on a stage. Therefore, the stage involves the presynaptic cell, the synapse, and the postsynaptic cell, as illustrated on slide number 7. You'll then introduce all the characters involved in our play of synaptic transmission using slide number 8. This slide is animated so you can introduce one at a time. Slide number 9 allows you to expand on our most common neurotransmitters and their functions. The key point of the activity is to allow students to discover the steps involved in the process of synaptic transmission. We'll get to this key point with a card sort and a Socratic discussion. In the activity, you'll give pairs of students a set of synaptic transmission signaling cards for them to sort. The 8 cards are written such that if students are reading carefully, they should be able to order them fairly easily. After giving students 5 minutes to complete this task, you use the next set of slides within the lesson PowerPoint to review the correct order of the cards, which describe the steps involved in synaptic signaling. At each step, you'll review and elaborate. For more information about each of the following steps, please see the teacher manual. The steps in order are: Number 1, the action potential invades the axon terminal. Number 2, voltage-gated calcium channels open. 3, calcium flows into the presynaptic cell. 4, calcium-sensitive proteins fuse synaptic vesicles to the membrane. 5, neurotransmitters are released into the synaptic cleft. 6, neurotransmitters bind to postsynaptic receptors. 7, ion channels open on the postsynaptic membrane, allowing ions to flow into the cell. And 8, excess neurotransmitters are degraded by enzymes or pumped back into the presynaptic cell. The key point of the wrap-up is to give an example of the effect of disrupting synaptic transmission. We'll get to this key point by examining how Botox works. In the wrap-up, you'll ask students what would happen if you took a drug that destroyed the calcium-sensitive proteins that fuse synaptic vesicles to the membrane. This question allows you to assess how well your students understand the process of synaptic transmission because it forces them to apply their understanding to make predictions. And basically what happens is that you wouldn't be able to release synaptic vesicles. Interestingly, that's how Botox works. By stopping the release of synaptic vesicles, it prevents muscles from contracting, which prevents fine lines and wrinkles in small doses. Large doses will completely paralyze you and thus kill you. The key points of the homework are to review the steps of synaptic transmission, as well as to predict the effects of modifying the process of synaptic transmission. We'll get to these key points by completing the worksheet. You'll use slides 19 and 20 to introduce the homework assignment, which has students review the steps of synaptic transmission and think about whether it matters which ion flows into the postsynaptic cell. Students often ask for more details about neurotransmitters and drugs that affect synaptic transmission. You can find more in the teacher primer, the teacher manual, and the student workbook. One of the most common questions teachers get is Botox the only drug that modifies synaptic transmission? Absolutely not. In fact, most drugs that affect our nervous system do so through modifying synaptic transmission. The drugs are, after all, chemicals, so it makes sense that they would alter a chemical process. Examples of drugs that modify synaptic transmission include caffeine, all drugs of abuse, which we'll get to in Unit 5, and medications, including antidepressants, anti-anxiety meds, and ADHD medications. At the end of this lesson, students should appreciate how neurons convert the electrical signal of the action potential into a chemical signal at the synapse. Students should also be able to use this understanding to make predictions about the effects various drugs would have on neuronal signaling. And finally, students should begin to appreciate how different types of neuronal signals, action potential, synaptic transmission, and dendritic input signals all relate to each other. This lesson builds on students' understanding of neuronal signaling, which includes both action potentials addressed previously in Lesson 2.2 and synaptic transmission addressed here. This lesson provides the foundation for future discussions of both inhibitory signals, which are addressed in the next lesson, and how drugs of abuse modify synaptic transmission, which will be addressed in Unit 5. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members, and we'd be more than happy to help you.